Antibiotic resistance has become a real threat to global health. Multi-drug-resistant pathogens are responsible for 700,000 deaths annually, and it has been estimated that this number could increase to 10 million by 2050, meaning that drug-resistant infections would be killing more people than cancer. New antibiotics need to be discovered that bacteria have not yet developed resistance to, but pharmaceutical companies are hesitant to invest as the process is so expensive and new antibiotics will need to be used sparingly in order to increase the length of time over which they are effective. This means lower sales, resulting in virtually no profit for investing companies. Therefore, a different solution is required. In a large bacterial population, there is lots of genetic variation as a result of random gene mutations and horizontal gene transfer. This is where genetic material is transferred between bacteria, but not vertically as it is from parent to offspring. By chance mutation, some bacteria will possess a gene conferring resistance to a certain antibiotic. These resistant bacteria are shown here in red. When antibiotics are administered, they kill the majority of the bacterial population by disrupting a key step in their metabolic processes. Some of the body's good bacteria is killed as well. However, the resistant bacteria survive and go on to reproduce, passing on resistance to their offspring and resulting in a majoritively resistant population. Resistance can be passed from bacteria to bacteria by horizontal gene transfer as well. These are just some of the causes of antibiotic resistance. A potential solution to the antibiotic resistance crisis is phage therapy, the use of bacteriophages, which are viruses, to infect bacteria in order to treat infections. Bacteriophages do not infect eukaryotic cells, which are those with a nucleus like human cells. They can only infect bacteria. The simple structure of a phage is shown here. They generally consist of an icosahedral head containing genetic material made up of nucleic acid, and a tail which ends in tail fibers that bind to receptors on bacterial cell walls, allowing them to infect the bacteria. The viruses infect bacteria by injecting their genetic material into the bacterial cells, and the viral genetic material is shown here in red. They also cause the bacterial genetic material to disintegrate. They then hijack the bacteria's reproductive machinery, forcing it to replicate the viruses and produce viral particles and proteins. Then when the bacterial cell lyses or bursts, it releases all of the new phages that have been assembled inside the cell. This shows the bacterial cell bursting, releasing all of the new phages that have been assembled. This process continues until the phages have infected all of the cells in the bacterial population. Many antibiotics are broad spectrum, meaning they target a wide range of bacteria and can even harm the good bacteria in your body. Phages are narrow spectrum. There are many different types which can each only infect one specific bacteria, so they don't harm the good bacteria as well. This is a bacterial cell. One of the most promising benefits of phage therapy is that phages can be used to increase the susceptibility of bacteria to antibiotics, essentially reversing resistance. An example of this can be shown using efflux pumps. One of these is shown in purple. Bacterial efflux pumps are membrane protein complexes which transport molecules from the inside of the cell to the outside. Some can transport antibiotics. Bacteria with efflux pumps specific to a class of antibiotic will expel these drugs and therefore be resistant. However, a particular bacteriophage binds to efflux pump proteins in the bacterial cell walls in order to infect them. Therefore, all of the bacteria which possess efflux pumps in a population can be killed by phages. The phages can't bind to those which don't have efflux pumps, and so these bacteria survive the phage therapy but are left defenseless against antibiotics. Now they have no way of expelling the drugs. If the bacterial population shifted to develop phage resistance, they would need to get rid of their efflux pumps, and this would hugely increase their antibiotic sensitivity. This trading of vulnerabilities principle can hopefully provide a new way for multi-drug resistant infections to be treated. In order for phage therapy to become a reality, research and development of new antibiotics, and in particular new classes of antibiotics, is vital. Alongside this, toxicity, safety and stability studies of phages should be undertaken to determine how safe phage administration is and how the phages can be stored for clinical use, seeing as they need to have bacterial hosts in order to survive. Of course, clinical trials would be a crucial step towards implementing phage therapy into general healthcare, and a few have already taken place. Larger trials will need to be completed before phage therapy can truly be considered a solution to antibiotic resistance. Finally, here are some articles showing the success of phage therapy in curing multi-drug resistant infections, saving the lives of 2 patients. One was a teenage girl with cystic fibrosis at Great Ormond Street in 2019. The Eliava Institute in Georgia delivers phage therapy to patients regularly, as phage preparations are licensed drugs in Georgia. However, it's difficult to know how successful this is as they refuse to publish a success rate. Obviously, further scientific studies still need to be conducted, but phage therapy definitely has the potential to be a successful treatment option in the future. Thank you for watching!